Okay, excellent. So I'm just going to start introducing, and meanwhile, students can still join, of course. So we have the pleasure to have Rosa Cossart talking today. She is the director at the Institute of Mediterranean Neurobiology, affiliated to INSERM. I hope I pronounced this all correctly, and Aix-Marseille University, a pioneering institute in the field of systems developmental neuroscience. After graduating in mathematics and physics from the École Centrale Paris, she studied the functional rewiring of GABAergic circuits in epilepsy during her PhD with Dr. Bernard and Ben Ari. As a postdoctoral fellow with Dr. Juste at Columbia University, she pioneered the use of calcium imaging to study cortical circuit function, and her lab made seminal contributions to the understanding of how developmental scaffolds hippocampal circuits. They discovered hub cells and more recently assemblies forming the functional building blocks of hippocampal function. And in this talk, she will present her experimental, or she will talk about the hub neurons in different cortical regions, I assume, if things haven't changed since. So yeah, we're very happy to have you here.
Thank you.
Yeah,
Thank
you very much.
Thanks a lot. Yeah, it's great. So I'm very happy to share with you this long story, like long chase of hub cells in vitro and more recently in vivo. So everything I'm going to talk about was done at INMED. So the institute that I now have the pleasure to direct, that is very close to the Mediterranean, not very far away from where Claire is now, and. So it's a fantastic, really a great environment, not only because there are the Calanques, but also because it's a highly interdisciplinary environment, as we are now also members of an institute called the Turing Center for Living Systems, which brings together experimentalists and theoretical scientists a little bit in the same format as the group you are today. So as you said, I'm an engineer by background, an engineer who became really an experimental neurobiologist, and I'm always trying to bridge the gap between these two aspects of my training. And so I thought that maybe the best example of such really a study that is really between theoretical and experimental neuroscience is this study of hub cells, and I will tell you everything we know so far about them from their birth to their role even in adult circuits. So it's a story that started more than 10 years ago, and again I think a nice example of how experiments can be fed by theoretical predictions, and also by methodology developments. I think that so in the title shedding light, there is light, and we have been very lucky to be able to use along all these years new methodologies to with light to try to dissect and know better about the organization of cortical circuits. So I'd like to start with this slide, because I mean, you asked me to tell you also about my background and my path. And so, as I said, when I entered into neuroscience, I was an engineer, and I didn't know much about neurons. And I even thought that neurons were all the same, that they were like a cable with yeah? Can you hear me? Yeah. Okay. So, and I like this slide because that's really when I started becoming interested in neuroscience. I was fascinated by the morphological diversity of neurons, and in particular those that are the GABAergic interneurons of the hippocampus. So this is a drawing taken from a very important review to me by Freund and Bujaki. And I was really, so it's a piece of hippocampus, and I must confess that really at the time, and for many years, maybe because of my background, I was more interested in the network in neuronal networks than in really the function of the area I was studying. But I already understood when looking at this diversity that there was probably like a division of labor among these different cell types. And that was an idea that was really very inspirational at the time when I entered into neuroscience. And obviously, when you look at those diverse cells, but nevertheless very stereotypical and characteristic, that's really something that was really inspiring. So, so networks of neurons, like other networks, in fact, of any interacting elements, they can be described in many ways, and in particular by their topology. And so what is a hub? One of the most commonly described properties of a network, as I said, is its topology, and in particular its degree distribution. So the degree being defined as, for every node in a given network, as the total number of links to other nodes. So the degree distribution is in fact often used to distinguish between different types of networks, including brain networks, like regular networks where every node will have an equal number of partners, or random networks. So a particularly interesting topology of networks are these so-called scale-free networks that are characterized by this power law distribution of connectivity degrees that you can see here. So what this power law means is that whereas most cells in those networks are poorly developed, there are a few cells in this heavy tail at the end of the distribution that carry an exceptional number of functional links. So those cells are called hub cells. They are again a small fraction of highly connected nodes buried in an otherwise really poorly connected sea of neurons. So they are obviously particularly appealing in neuroscience because coordinated activity, so the capability to influence the activity of many other cells in a given network, is really a hallmark of most brain operations, from development to complex cognitive functions. And I will just illustrate that in 2 slides. So in the development...
Rosa, I have
yes?
a question.
Yeah, sure.
Would you say that the scale-free, like a network, are the network that we most commonly find in the brain or cortex,
so
or in most naturally occurred network in general, not maybe only in the brain?
I mean, that was a theoretical prediction. It was also based on analysis of simple networks like the Drosophila, or like based on connectivity. It's also a very often found connectivity if you in like fMRI studies, so networks of interacting brain areas. Yeah, but just one thing, I'm not sure. I mean, I'm always hesitant. So I use this slide because I want to start making, like, with the terminology and making things clear. But in a way, maybe it doesn't matter so much that the network is scale-free. I think what is really important is the fact that there are indeed a few exceptional cells that have the capability to coordinate the activity of many others. So yeah, and so scale-free networks now in adult cortical brain circuits, there are also evidences of scale-free modes of organization. The fact that most cells are inactive also in adult cortical circuits is also an indication of probably a scale-free type of topology. But yeah, I don't know how universal it is, honestly. I mean, it's yeah.
This scale-free characteristic in network is like a part of a project that I did do, like yes. building a like just completely network, not neuroscience related, but just building a model with random walker, like the growing model.
Yes.
And then we analyze the model, and we do see a very clearly scale-free, scale-invariant characteristics of the model. That's why I'm curious.
So yeah, and but in fact, so in a way, so as you know, one way to build a scale-free network is through this preferential attachment rule, by which a new node in the network will preferentially connect to one node that is already heavily connected. And when you think about that, and it's exactly the prediction we've tested with hub cells, it's like something that is quite natural. I mean, it's something that happens in the brain. You have cells that are going to be there first, and they will connect to cells that they will be the most connected cells. And from that growth process, it is quite likely that at the end of the day, we will find a scale-free pattern of connectivity. Now, there are structures where I like, for example, the cerebellum, where I wouldn't say that the connectivity is scale-free. So probably it's something that is, but still, given this fact that if you just take this simple rule, or even less, I've seen that there was a paper that showed that even the preferential attachment rule could be just a correlate of so-called homophilic type of attachment, where nodes will preferentially connect to nodes that are similar to them. And if you take also just this simple rule, you end up with a scale-free pattern of organization. So yeah, I mean, it's still, yeah, I don't know how much it can be generalized, but yeah, it's, so
sorry, can I also just quickly ask?
Yeah, sure.
It's so early on.
That's good. That's nice.
I wanted to ask, you said like nodes might connect to like nodes that are similar to them. Like with the hub neurons, do you, like, could they be distinguished just on like transcriptomes? Like, or is it mainly the connectivity? Like, do they also show similarities on a transcriptome level?
So that's something I'm gonna, I'm gonna touch upon the identity of those cells, whether they are specified, but yeah, so I will tell you a little bit more later. But it's also, I mean, those are in a way still open questions, because yeah, is a hub cell determined by its genetic, is it genetically programmed with specific features, or is it just the consequence of a growth process and of being like just the first cells to be there? I mean, that's still, both are still possible. And when you'll see a little bit more about the identity of hub cells, maybe these different alternatives can be better discussed. So yeah, so just, I mean, so we care about hub cells, for me, like, because it is always a challenge to understand how activity can be coordinated in the brain. And as I said, most brain activity patterns are really coordinated activity patterns. And so finding cells that could be playing a key role in organizing the activity across different cells is obviously an important question. And so just one example, I mean, you all know that, but but I was thinking I was speaking to people that didn't know anything about the brain. So just one slide to tell you that coordinated activity patterns are really a hallmark of all developing brain circuits in different species, like they can be recorded in vivo, in vitro, with calcium imaging, as you've seen here. So those are really like before active exploration of the world, most developing brain structures are active in these recurring bursts, synchronous bursts of activity. So here you see them nicely on these raster plots. So, and these activity patterns have a role in the maturation of functional brain circuits. So they are not just an epiphenomenon. Now, they are also important, these coordinated activity patterns in the adult brain. And I'm using the example of the hippocampus. So in the hippocampus, that is, so it's a brain area that is important for memory processes. What I find particularly amazing is that there are these spontaneous peaks of synchronous activity in vivo, in the adult brain hippocampus, that have a role in the, that are implicated in the memory function of the hippocampus, since they reactivate patterns of neuronal activation observed during experience. So these, and really they can be detected, these reactivation events, as just synchronous calcium or synchronous neuronal activity events. So again, the possibility that some specific cells could be playing a role in coordinating these reactivations that are important for experience-dependent plasticity is particularly appealing. So what you may also see here is like I was mentioning that is the difference in the level of activity between the adult and the developing brain. The adult brain is much more silent, but even though there is within only a minority of cells, there are these particular events that are, that are important again for contributing in storing the experience. Okay, so the question obviously that is really the main question throughout this talk is, are there indeed hub neurons coordinating cortical dynamics during development and maybe later? And I'd like to start with this slide. So I will tell you a little bit about the methods, because I think methods are always important. So of course, the question is important, but sometimes even you can, just by using like novel tools, you may even find things that you were not expecting. And so to me, something that has been really a big change in my scientific career, and that happened in fact quite early in my career, is the possibility to follow neuronal activity by looking at the calcium fluorescent dynamics in cells using microscopy. And when I'm saying that, because, so I told you I was first trained as an electrophysiologist, fascinated by neuroanatomy. But then, so with that, I was able really to go and perform a fine level description of single cells and their how they were embedded in specific circuits. But obviously the big picture of the population dynamics was not easily accessible. And when I was a postdoc, after my PhD, I decided to not completely abandon electrophysiology. I think electrophysiology is always a very important readout of neuronal activity. It's even more important now, because I think that a lot of people start using calcium without any experience of neuronal physiology, which may be problematic. Okay, but nevertheless, I mean, really, calcium imaging has been really a game changer, because as you can see from this slide, it allows really a quite good mapping, tracking of between the neuronal spiking and calcium fluorescence transients when you have a dye in the cell. So here, I'm using here an example. So this is something, a calibration we did in vivo that shows really clearly how neatly the calcium fluorescence changes one can observe in vivo in a cell, can nicely track the underlying action potential dynamics. So those are single spikes recorded in cell attached mode while imaging the same cell. And you see how when the cell fires bursts of spikes, you have these larger calcium transients occurring. So from that, I have, so it's something I started as a postdoc. I designed an experimental approach that is really, that can be nicely applied to the chase of hub neurons. So that links these population dynamics to the behavior of single cells through this loop. I have this little picture here because, so that was something that, as I said, I started as a postdoc in Rafael Yuste's lab. It was like almost 20 years ago now. And with someone that has been really instrumental in setting up this approach, and his name is Dmitry Aronov. He's now an established professor, he's an assistant professor at Columbia University, running a lab where he's studying spatial cognition in birds. But the reason why I took this picture is because most of the important moments in my career and important findings happened when an experimentalist was working hand in hand with a theoretical scientist or a computational scientist. And Dimitri, at the time we were working together, was a computational scientist. He was not yet into neuroscience, and I was the experimental neurobiologist. Okay, so this method goes, as I said, from the looking at the spontaneous or the ongoing calcium activity in the brain. It uses methods to extract functional connectivity degrees online. So while the brain is still alive, or the cell is still alive, and then identifies the nice functional connectivity motifs, for example, the highly connected hub cells. And we then go, and I think this is also something that is really super important, is to go from this observation to the manipulation of the specific cells or patterns we are able to outline from the analysis. The reason why I'm saying, so I think that, and we do that through electrophysiology or optogenetics. And if we do it with a patch pipette, we can also resolve the neuroanatomy, so the specific properties of that particular neuron that was responsible for this particular functional connectivity motif. Okay, so I think I want also to insist on this point. I think that it's nice to look at what cells are spontaneously doing before starting to use light to manipulate their activity. If we do that without this prior observation, we may reveal something that is not spontaneously occurring in the network. And hence, again, I insist, I think that really going from spontaneous activity to evoked or if probing or light probing of effective connectivity is an important step in this process. So functional connectivity, so that we are just on the same page. I mean, it's super easy. So there are many methods, and I'm sure among you, maybe some of you know about more fancy methods to infer functional connectivity from neuronal activity measurements. But still, so far, our approach is based on pairwise correlation of activation onsets, as you will see from this slide. So whenever a pair of neurons is active, we build for each pair of imaged neurons these little histograms that plot the distribution of the spiking of neuron B centered on the spikes of neuron A. And if there is an average positive difference in the way neuron B spikes with respect to neuron A, we say there is a direct functional link from A to B. And the other way around if the difference is negative. And if there is a flat distribution, we don't consider any link between A and B. And yeah, so this is based on, so we have statistical tests to prove that indeed there is a significant positive difference in either direction. What I should tell you also, I think this is important, is that calcium indicators, we don't claim that these are direct monosynaptic connectivity patterns. And also we have to exclude cases where the spiking of neuron A and B are perfectly correlated. If they are centered at 0, these histograms we discard we don't say that there is a functional link between A and B. So we probably underestimate the level of connectivity of our networks. We only describe those differences in dynamics that are statistically different. Okay, so
Hey Rosa, can I ask
yes.
you a question on this?
Sure.
Did you ever try to on the previous slide to actually correlate with mapping connectivity in slices? Just out of curiosity in preparation where you would
yeah,
increase?
so it's something we did in fact in our early study with so here when we looked in slices, so that was we have and I'm really, so that's something, it's good you asked the question, because sometimes your referee number 3, you know, the one that is really making your life difficult, can be the one that makes you do the difficult experiment that once it's done, you're happy about it and you are much more, you know, much more confident about what you say. And so one reviewer, so when we were doing that, so going from the analysis of spontaneous activity, so here we are in the developing hippocampus. These are slices of mouse at around P7. We did this analysis. We found this so kind of scale-free degree distribution, found highly connected hub nodes, and then patched them. And the reviewer was asking us, okay, and we found that the cells had an incredible morphology. So in our initial submission, we said, okay, we have this perfect correspondence between anatomy, recovered with neurobiotin fields, and functional connectivity. So most likely, hub neurons have this amazing impact on network dynamics because they are structurally very well connected as well. But the reviewer said, but you should show it. And so this meant we had to do targeted, because it was before optogenetics, okay? So we had to do targeted double patch recordings from, for example, this little red cell and one of its functional targets. And we did that. So it's a supplementary figure in our in the Bonifazi paper. But it's a beautiful finding because not only, so we find that, so there are several things. In fact, it turns out that many of the postsynaptic targets we found were GABAergic cells. So, and they had a direct, really a direct synaptic link to the hub neuron. So there is not all of these functional links are going to be through direct monosynaptic connections, but a significant fraction of them is indeed through those direct synaptic connections. So there is, so it's...
And do you have any evidence that the hub are connected among themselves?
So this is also, I mean, this is something we have not really done. So I think I'm going to, so we have on the, in the in vivo data, we found that they had, they displayed a kind of rich club topology. So on regarding functional connectivity, they seem to be more connected one to another than to other cells, okay? So they have, but these are functional links again, so it doesn't mean that they are more connected through direct synaptic interactions or gap junctions. That's another possibility. So yeah, so it's a really, in a way, this so this finding of hub cells, so we found that they were GABAergic. I'm not sure. Yeah, so I will show evidence later. And so this is a typical, not a typical, there is no typical morphology, but the typical thing is this amazing widespread axon. So they are here in red. So this cell was patched. It was patched by me, this one, I know. So I did that experiment. It was filled and reconstructed. Yeah, I mean, if I can say something, I mean, you guys are still young because you're graduate students, right? But I think that once you become a young PI, it's also quite important to continue doing experiments in your lab and for a while, because if you leave the experimental ground too early, I think then I mean, having you as an experimenter is really boosting the lab a lot. And yeah. So we were, so I don't know if I did this experiment, but I like to show it because it's like, so you see the patch pipette, it was our finding of hub cell was a little bit too early, because as I said, it was just about the time when optogenetics was kicking in. But before, so we couldn't use light to stimulate those cells, and we had to do this very difficult targeted patching in so find that cell among hundreds of cells in a living slice was difficult. So here it's a hub cell, sorry, I should, and we are, so this is the cell is first held in current clump at its resting membrane potential, it's spiking. And on the bottom, you see the fraction of active cells and the function of time. And you see these peaks in these histograms. They are the spontaneous bursts that are important for developing circuits. So in a minute, we bring the hub neuron to, in a minute, just now, we are bringing the cell to a different membrane potential where the cell is firing like crazy. And what you see, I mean, if you look at the movie, you can see that cells are still blinking. So, and believe me, it's the fraction of active cells is still the same. What disappears is these coordinated action bursts of activity. So, and then we bring back the cells to its normal. So it's a real experiment. So things are not optimal. And you see that there were some hesitations, but now the cell is back where it was at the beginning, and you see that the synchronizations also come back. So that's quite convincing to us. I mean, those experiments were really like, when we saw, there were not so many cases where we could see really with our eyes the impact of stimulating a single cell on the network, but this was one of them. Okay, so this was in vitro. And my lab, I must say, for many years was really mostly working in vitro because, as I said, I was more fascinated by the behavior of the networks than really by the possible relevance of that network behavior to any kind of function. And but then we decided more recently, and it's in fact still the paper is still in revision, but hopefully, I mean, I hope it's going to come out soon. So we decided to probe the existence of hub cells in vivo. And I must say that one of the strongest drive to really do all these experiments again in vivo was because we thought that, so we knew that those coordinated bursts of activity, they are strongly triggered, influenced by sensory motor inputs, which obviously are not there in a slice. And so we wanted to see how this particular network topology that, I mean, it's there, we find it in a slice, how it translates in vivo in circuits that are also receiving this sensory motor information. And so again, so this is the second picture with, so as I said, I should have said that before, but the hub cell, sorry, let me go back, but the hub cell story was again the product of this amazing combination of Paolo Bonifazi, a theoretical neuroscientist, with Miri Goldin, who was an experimental neurobiologist. And in fact, they are even a couple and I have 2 kids together now, but they met on the hub cell study in the dark room of the in vivo to photon microscope. So, and then later, so more recently, Laura Modol, who is an experimental neurobiologist now in Oscar Marin's lab, and Yannick Bollman, who was a computer scientist, also teamed up to chase for hub neurons in vivo. And this was again a nice thing to do, to pair these two. And so we did, I mean, the same thing as in vitro. We calculated functional connectivity decrease the same way based on pairwise activity correlations, and looked at how these connectivity degrees distributed. So I wouldn't make a big claim about the scale-free organization of these in vivo patterns. In fact, they are probably more exponential than really scale-free. But nevertheless, there is a really, there is a region where a few rare cells have an exceptional connectivity degree, and those cells only emerge after the end of the first postnatal week. So that was something that we didn't really know. As you can see here, so those graphs are plotting the most connected components. So every cell here is connected at least by one link, and the size of the circles in these graphs depicts the number of connectivity degrees each cell has. And you can see that after P7-9, we start seeing those cells that are among the 5% most connected cells in these developing networks. And what I forgot to tell you is that we probed for the in vivo existence of hub cells in the barrel cortex for several reasons, and one of which is the fact that it's much more accessible optically than the hippocampus. And also it has a very nice direct sensory input through the whiskers. So the hub cells are so highly connected cells, as I said. So this is the quantification again, showing that only after P7 we see the emergence of these highly connected nodes. And we have also a significant increase in connection density as development proceeds. So we wanted to do the same type of experiments as the ones we did in vitro, and for that we had a very nice collaboration with Valentina Emiliani and a joint supervision of a PhD student, Thomas Tressard. And the collaboration was about using holographic stimulation to target the cells that were candidate hub cells to see what was indeed their impact. If we could reveal, go further from the description of functional connectivity to the description of their effective connectivity, so their causal impact on local network dynamics. And so this took us this was really hard work. We spent more than maybe 5 years building a setup. And then when we tried stimulating the cells, we failed first because it was impossible to induce a significant cell response using the classical obscenes. What you need to understand is that performing calcium imaging together with optogenetic stimulation is really a challenge. There are several options. You need to avoid stimulating the obscene while you are scanning the network for calcium imaging. But nevertheless, you need to have a sufficient powerful enough obscene to activate your cell. So those were really, and I must say that it was more like a cooking problem than really a very hardcore experimental or engineering issue, because the solution to our problem came when we were able to use these fast obscenes and soma-targeted obscenes, Chrome, that were developed in Hillel Adesnik's lab. And with those obscenes, we could really efficiently target cells with light in vivo. So this is just an example of, like, you can see here how neatly the obscene is expressed only in the soma of the cells. And since this is the obscene is delivered through a virus, so it's a flexed obscene, we can choose whether to induce obscene expression either in GABAergic cells or in glutamatergic cells, okay? And so, yeah, so you can see here that when we patch a cell and record the response to the light stimulation, these obscenes are really very efficient in driving action potential in the cell at reasonable power. So, so I'm showing you so the typical experiment. So we image at P9 since it's the time when we start seeing hub cells. So again, so here we are using JCOM to map calcium dynamics. And so whenever a cell is active, it gets brighter. So these are the typical ongoing dynamics in the barrel cortex at P9, same thing as in the hippocampus. And if we plot the pattern of activation of every cell as a function of time, we see these nice the synchronous patterns of activity. So what we do is that we analyze the network, the ongoing dynamics, and find candidate cells for stimulation, such as this cell, which has this high exceptional number of functional links. And we then, if we are lucky enough, the cell also expresses an obscene, so it's it can be stimulated by light. And so this is an example of, so we are now stimulating the cell. You can see that, you can see the stimulation artifacts. You can see that the cell gets brighter during stimulation. And well, on this example, so this is the response of the cell to the light stimulation. And what you see is that indeed there is a significant change when we stimulate the cell, and it's reversible when we stop stimulating in the what we call interim event interval, so the time interval between two consecutive synchronous network bursts. So those, so this was a case where we were stimulating a GABAergic cell. I'm gonna show you a more spectacular example. So here the hub cell is located here. It's also expressing the obscene. It's also GABAergic. This is the baseline movie. So cells are, again, spontaneously active together in a recurring manner. And we will soon start stimulating the hub cell. You will see the stimulation artifacts. And you see the cell is very bright. And you maybe see, I think it's quite clear. I showed a movie to my daughter this morning, and she could see what was happening. So the network is really much less synchronous. And if you miss that, when we release the cell from stimulation, you see how synchronization comes back. And so this is, of course, highly significant. So those are really.
Rosa, sorry.
Yes.
Did you try to play with other GABAergic neurons that wouldn't be hub neurons?
Yes. And many. So yeah, I don't know if I have the slide showing this quantification, but in fact, the problem is that most cells don't have any network, any impact. So it's we sampled a lot of cells, and none of them has any significant network impact. We have even among the functionally connected cells, so it's among the highly functionally connected cells that we have a network impact, okay? But it's not a 100% thing. So it's not all the highly connected cells that show this behavior, but none of the poorly connected cells has any significant effect on the synchronization. The same, and we've tried also stimulating, why doesn't it, voila, highly connected glutamatergic neurons, because so the property of displaying high functional connectivity degrees is not specific to GABAergic cells. You have also glutamatergic cells that are highly functionally connected, like this one. But when stimulated, so they respond very nicely, they express the obscene, but we could not find any significant network effect. So yeah, we have as many controls as, I mean, we have many more control cases where nothing happens than cases where something happens. And yeah, and the nice thing is that things happen in those GABAergic cells that have more functional links than the others.
I have a second question, actually. The pattern of stimulation that you use, could it be releasing also peptides from the GABAergic neurons, or do you think it's just GABA?
So that's a great question. So the pattern of stimulation is quite. So we stimulate. We give 10 pulses separated at 10 hertz. So it's quite a long stimulation. So indeed we could release other things than GABA, and it's and in fact, it may be even, so I'm not going to talk about that, but work that we are doing in the hippocampus suggests that some of the hub cells that are somatostatinergic may in fact have an influence on the development of GABAergic circuits through their release of somatostatin. So yeah, I think it's a good thing we shouldn't forget that. Maybe it's not everything goes through GABAergic transmission. Yeah. So as I said, one thing that was interesting is
Sorry,
yes.
I also have a question.
Sure.
It's not directly related, but can you, when you look at the neurons at that point, can you figure out how old they are? Like which ones are older or is there a technique?
So yes, so this is something that I'm gonna touch upon later, but I can tell you already. So we have done in a separate set of studies what is called inducible genetic fate mapping. So we have labeled cells according to their birth date, and we use transgenic mouse lines that express a specific set of transcription factors. For example, well, mainly DLX1-2, that is important for the that starts being expressed in interneurons when they start migrating. And so if we give tamoxifen pulses at specific time points during embryogenesis, we can, with this method, tag neurons and make them express a dye throughout their life according to when they are born. And so that's something, I'm going to tell you something about that quickly in a moment. So just one thing, because about as I said, one of the main reasons why looking in vivo was an important endeavor is because there is this link between local circuits dynamics and sensory inputs. And so we first looked at the impact of whisker stimulation on our image neurons, and what we found, and I should have added a quantification, but I'm just showing an example. So here we are baseline, and then we start stimulating. I mean, it's not a very fancy whisker stimulation. It's something really very unspecific and quite broad, but and what we observe is that those cells that respond the most to the whisker stimulation are also cells that could qualify as hub cells. They are highly connected, whereas other cells are not responding as much. In fact, the whisker stimulation at P9 tends to inhibit the network more than anything else. We have also looked at sensory deprivation. So here we plucked the whisker for a few days after birth, and then looked at the development of the networks, and what we observed is that the emergence of those highly connected hub cells that, as I told you, starts at P9 is prevented or delayed if we remove these sensory input. So, and that's really quite striking and strong effect. Now, I'm coming to the slide that I was advertising when I started. So these are experiments that we did. So it's like the experiment that reviewer number 3 asked to have this study published. Well, hopefully it will be enough. In fact, it's not even the reviewers who asked, it's the editor, which yeah. So, so we have this indirect link between hub cells and sensory inputs. We have also this link between hub cells and local circuits. And how can we directly probe the impact of the relay between sensory inputs and local circuits through hub cells? How can we show that hub cells are really important in relaying sensory inputs to local circuits? So one way to do that is to do the same whisker stimulation protocol as the one I was showing earlier, but this time while inhibiting hub cells, okay? So we've done those experiments only with GABAergic hub cells. They are quite complicated experiments to do, and but so this time we express a soma-targeted inhibitory obscene, again from the Hillel Adesnik's lab, and it's been working very nicely. So this is a global average trace of the response of the photoinhibited cells. So there are 9 cells here that are photoinhibited, and you can see here the kinetics of the response. So this is one experiment where I think you kind of see something. So, so this is the baseline activity. Again, you have these vertical lines typical of synchronization. Then we start stimulating the whiskers. As I told you, the whisker stimulation tends to kind of silence, in fact, the ongoing network synchronizations. And this is something you can, in fact, see here in this measurement. So here we are measuring both the amplitude and the frequency of spontaneous synchronous calcium events. And so then after this period of whisker stimulation, we start whisker stimulating, but while photo-inhibiting the hub cells. And what we observe is that we almost return to a baseline. So as if inhibiting the hub cells was preventing this whisker-induced inhibition of local circuit dynamics. And what this plot is showing is that those cells that have this strong network impact when photoinhibited also have a higher connectivity degree. So again, saying that what I'm saying here is that when you specifically photoinhibit cells that have a high functional connectivity degree, you modify the response of local circuits to whisker stimulation. Okay, so that's quite a direct link between hub cells, sensory inputs, and local circuits. And so what we think now, I mean, it's things that we can discuss if you want later, but that really hub cells are important nodes in calibrating the local circuit dynamics to the statistics of external sensory inputs.
I have another question.
Sure.
I feel like there are probably two, but I'm just gonna start with the. Do you think, because for whisker stimulations it makes sense because you would want to have coordinated activity following the sensory input, but would you say it would be the case for all sorts of sensory input? Let's say some where you would not want to have this constant coordinated activity as it might drive changes overall that, like, you would kind of try to prevent. Like, do you think there would be some kind of mechanism that would stop these hub neurons to always allow for this coordinated, so like some kind of filter before, let's say, or something
you're meaning that there would be, if the sensory input was not, so whether we try to look at the impact of other sensory inputs, that's the
yeah, basically like sensory input that might not be kind of natural in a way, because like for like natural sensory input, like you want this like feedback, but if there's, yeah, I guess something.
What you're saying is that the stimulation protocol that we are using is not a natural sensory input because we are, and that's the reason why the network is inhibited by the sensory input, or no?
No, actually the opposite, but maybe it doesn't really make sense. No, like I kind of, yeah. So like if you have the sensory input and it's kind of relayed by the hub neurons to then allow for this like local, like kind of synchronized activity and like kind of further downstream, which probably has a higher impact in the end overall. That's, I kind of saw it as like a relay that allows for like a bigger impact later on. And in some scenarios, you might want to avoid this like bigger impact instead of having, I mean, it is inhibited like overall, so I guess it's fine, but instead of kind of, so like I wondered whether there are some sensory that might you want to avoid going via the hub neuron. So instead of having everything kind of coordinated, kind of want to bypass this mechanism because you don't want this coordination.
Yeah,
I don't know.
Maybe those are experiments that can be done. I mean, I don't know what I would really what I would expect from like, I must say that these experiments of the whisker stimulation, they have a strong circuit ground. Like they are inspired also by what we know about the development of the barrel cortex, circuits in the barrel cortex, and there is in fact really a specific and transient link between the thalamus and GABAergic cells that are located deep in cortical layers.
Okay, yeah, I was kind of thinking of the thalamus as well. Yeah.
So there is really, like, I think, so behind this functional observation, there is also an anatomical basis for this mechanism, like of this route of information flow. So now about, yeah, I agree that the type of, it may be that there are conditions where hub cells would function in the other way, and that would be interesting to see, like if you have a expected or like a self-triggered sensory input versus an unexpected one, it may be that hub cells transmit information in a different way in both conditions, and I think that's very important an important question to address.
Okay, thank you. Yes.
I have another question, if you don't mind.
Sure.
In this whisker stimulation experiment, did you try, or would you consider trying entirely ablating the cells instead of photostimulating them?
So yeah, in fact, we considered it because we thought we wouldn't be able to do that. So to, like, you mean like, for example, photo ablating the cell?
Photo ablating, for example, yeah.
Yeah. So yeah, we have not. We thought that the nice thing about the photoinhibition was that it was reversible,
yeah,
and more controllable. But to be honest, these experiments that we just, that we did like the beginning of this year, they were very complicated to do because the main authors of that study are now both in London, and due to the pandemic, it's like impossible for them to travel back to France, and the setup is only present here. And so, but they had spiral stimulation type of microscopes that would not be sufficient enough, according to Valentina, to photoinhibit the cells with single cell precision, but to photoablate a cell. So we discussed a lot about that. Yeah. I think that there is still room for experiments. There is room for many experiments, but for experiments where we would really see the long-term impact of killing hub cells on the development of circuits, and in a spatially and temporally controlled manner. So that's something we still hope we can do one day. And maybe using more like genetic type of approach to select hub cells. And that brings me, in fact, to the last. I mean, if I have, I don't know how much time. I think I don't know. Do I have a little bit more time? What's the?
Yes.
Yes. Well, I can be quick, but just to touch upon that. Those questions I was asked about the temporal origin of the cells. So we have shown that hub cells are very important around the end of the first postnatal week. What I'm not showing you is data indicating that they may be really important for the coordinated emergence of somatic inhibition, but I don't have time to tell you about that. What I just wanted to share with you is the fate of those cells in the adult circuits. And this is work done by Marco Bocciu and Pierre Gouni. So here we are not, it's not a combination of theoretical scientist and an experimental one. They are both experimental neurobiologists, but very talented ones. And so, so for that, we used the approach I was mentioning earlier. So an approach that enables us to tag cells according to when they are born, using these transgenic mouse lines. So it's something we have already published a while ago. And with that approach, now 10 years ago, Michel Picardo, who is now a permanent PI in my lab, had found that he had found the embryonic origin of hub cells. So we compared early born GABA cells to late born GABA cells and their network impact. Again, this was done in slices. And what he found is that those cells that had the earliest embryonic origin were the cells operating as hub cells with a network impact when stimulated, whereas cells born later were ineffective. And so what we found also, so I'm just summarizing the results, but is that these early born GABA hub cells, they stay in the adult circuits, which was not a priori expected, because a lot of pioneer cells, like subplate neurons, chiral rhesus cells, a lot of them are there transiently and then disappear, but those cells stay. And they become an interesting subtype of GABAergic neurons. They are not really typical interneurons, but interneurons that send axons outside from their main brain region, so outside the hippocampus here. They are long-range GABA neurons. So it's something we have shown using tracing methods as well as combinatorial immunohistochemical analysis. So they are a diverse population. They become a diverse population of long-range GABAergic neurons with also specific physiological features, including a tendency to be less intrinsically excitable than their control peers. They have also...
Sorry, Rosa, can I ask a very naive question outside of your field?
Yes.
So compared to the cells that Hannah Monnier is excited about, is it the same neuron that you guys are into or not?
It can be the same. Yeah. It's very likely to be the same because, for example, so here I'm showing examples of septal retrograde labeling of hub neurons, but we have also found, of early born GABA cells, I should say, but we have also found like injections in the entorhinal cortex can also retrogradely label those cells. So they can target different regions, and they can be also long-range GABA cells in the entorhinal cortex. So early born GABA cells, we found them to be long-range neurons in different areas, the hippocampus, the septum, the entorhinal cortex. It may even extend to other areas. So it seems to be an almost general property of the cell. So I would say that indeed, so Anna is very interested in the long-range GABAergic communication, and I would say that one way to label these population of cells is through this genetic fate mapping approach. Yeah.
So can you just say again where the somata are located?
So here I'm talking about hub cells in the CA1 region of the hippocampus. Okay.
They are also in other brain regions?
Yeah. So we found. So let me be specific. So we found functional hubs, so like what I described in the barrel cortex or using light, okay, we found those cells, we found them in entorhinal cortex, in barrel cortex, in CA3, in CA1, okay. Now, in CA3 and in CA1, we found that those functional hubs were comprised early born GABAergic cells, okay. So, then when we look at these early born GABAergic cells, we find that they are long-range projection interneurons, okay? We find that in the hippocampus, so cells with a cell body in the hippocampus that target septum, retrosplenial cortex, entorhinal cortex, subiculum mainly. But we find also that early born GABA cells in other areas of the brain, like the septum, or like the entorhinal cortex, are also long range. And this is because we've done some retrograde, at least long range to the hippocampus, because we did also retrograde labeling with injections in the hippocampus. So we were very excited. It's something we kind of dropped for a moment, but we were excited about the possibility that early born GABA cells form a brain-wide network of long-range connecting interneurons. Okay, but I mean, we have pieces of evidence indicating that this may be the case, but it's not like a complete set of evidence. So this is still open. Okay, but going back to this idea of identity versus like self-organized mechanisms of network organization, I think that, yeah, so they have this specific neurochemical identity. What I should say is that many of them express somatostatin, for example, but they are diverse in another sense, because they are not only targeting a specific region, they are targeting the various types of extrahippocampal areas. So they develop characteristic intrinsic features. So that's one thing. The other thing is also that they develop specific wiring. So I'm going a little bit fast, but the take home is that they receive fewer local inhibitory inputs than other control interneurons. And this is something we've shown using both immunohistochemical analysis. Like this is an example of a PV staining in a control GABA cell versus an early born GABA cell. They are almost here devoid of any PV contacts. It's something we probed also using IFEs, so they have less, they receive less iPSCs than control GABA cells, and they receive also fewer long-range excitatory inputs. So this is something we have probed using optogenetic mapping of the extrahippocampal inputs onto these cells. And I'm showing just here an example of light-induced stimulation of axons from the entorhinal cortex and from the thalamus, and from the reunions. So those cells are, as you see here, whereas most control GABA cells respond to the light stimulation of those inputs, the EB-GABAs do not. So they are...
Do you have any idea that they might express different factors to project long distance like they do, like during development? Do you think that they actually maybe express different factors to grow axons going far in the brain?
Yeah, so I still don't know. So one appealing possibility would be that they migrate by leave their axon where they are born. So it's, the septum is also a place of neurogenesis. So it could be that that's what happens, and it's something that we eventually should test. About their specific, about the specific tools these cells may have that make them different from other cells. I'm still hoping that someone will do like single cell transcriptomics. Like now it's accessible, we can sort the cells and analyze them, and maybe this kind of analysis will be informative to address the question you asked.
Or maybe patch-seq, right? If you guys can patch them, you could do patch-seq.
Patch-seq would be even more elegant, yeah. But yeah, we could also sort the cells based on their, like all the early born GABA cells, and see what.
What is the marker you use for early born GABA cells?
Sorry?
What's the marker you use for early born GABA?
We can anything you want. So we have transgenic. Because they are, so the markers. So with these, I should have included it. So with DLX12 CRE, so we have an inducer mouse line that enables the expression of whatever marker you want, TD tomato, GFP, channel rhodopsin, on the early born GABA cells only. So that's how we mark them. So yeah, so it's not a specific, we can label them with this approach, but it's not a labeling based on their identity, it's a labeling based on their temporal origin. But they are genetically accessible somehow through this method, which has the biggest limitation to be a method that allows to manipulate them only at embryonic stages. So it's not, so only using reporter mouse lines and not with viral approaches, it's still experimentally challenging. So maybe one last couple of slides about what they do in vivo then in the adult hippocampus. So this has been horrible. I mean, it's been a horrible project because those cells, what I should have said, but I mean, it's expected by the fact that they are hubs, they are very sparse, so there are very few of them. And for example, this is a lucky case of, so here we are imaging the CA1 pyramidal layer. This is a hub cell labeled in green, and there is only one. And this was really, again, a case where we were very happy to find one. The second thing, but I'm not going to go into the details, but this is something you have to know. It happens that transgenic mouse lines are not working the way you think they should. And this was the case for this one. If we were driving the expression of our reporter protein with a too strong reporter mouse line, we would have unspecific labeling. So it meant we could only look at hub cells with a GFP, so a green reporter, which meant that we had to do the calcium imaging with a red indicator. So GR Gecko, that's what we used. So which was okay, but not optimal. So so the hub cell is here. And this is the spontaneous activity in the adult hippocampus. So the hub neuron is quite active, and indeed, in a nutshell, that's what we found. So maybe I won't, yeah, I mean, I'm not going to go into that. I'm going to skip and go to the main findings. So we looked at how hub neurons were recruited, both during spontaneous run as well as during rest periods. Rest, both are important. During run, spontaneous activity, not spontaneous, but activity in CA1 organizes into sequences of neuronal activation. It's something that we have described in previous papers. And during rest, activity is organized into these bursts that I was mentioning at the beginning of my talk that are reactivation events, so events that reactivate the patterning of activity during experience, okay? And so we looked at how those hub cells were involved in both and found that they were highly recruited by locomotion, and they were also quite often recruited in these synchronous calcium events. Almost more than expected by chance for the synchronous calcium events, and they are highly significantly recruited during locomotion. And yeah, this is wrong. And we found also, we looked at their functional connectivity degree, again using the same type of approach as the one we used during early development, and found that so in a subset of field of views where we could image early born GABA cells together with principal cells, so in the stratum pyramidale, we found that the connectivity of hub neurons, I mean, it varied, but as a population, they displayed more functional output links than control neurons. And as expected from that, they qualified as hub cells as well in a significant number of cases. In fact, 1/3 of the hub cells of the early born GABA cells maintained this hub function in the adult hippocampus. And this is quantified here. So this is where they are. So they are, it's a 1/3 of them. It's not more than expected from the global population analysis. It's at the limit of significance. I don't know, maybe we should do more experiments to increase significance, or I mean, maybe that's just the way it is. So, so that was a long journey. I told you many things about hub cells. I think that really what we learned a lot about looking at them in vivo, because we start this in vivo chase for hub neurons really enables us to make interesting predictions about their role given their privileged connection to sensory inputs. The fact also that they emerge really quite abruptly between P5 and P7, a time when we see also the emergence of somatic inhibition. I think this is really something quite remarkable. So there are many more open questions, obviously. What I told you also is that those cells were not any cell. They were not among glutamatergic cells. They were GABAergic cells, and a significant early born GABAergic cells were part of the hub cell population. There are many, probably cells with other developmental origins that could also qualify as hubs. I mean, this is something we still don't know. And they remain in adult circuits where they display specific connectivity schemes, specific morphophysiological activity, and they seem to be bound to local dynamics in the adult brain, which is, I think, also very interesting if you consider their early developmental role. So that's it. So that's I mean, this has been like a very, very long development research project. I mean, it's still ongoing, and many people contributed to it. Even the people that were not directly involved really helped, because there were a lot of very difficult experiments, and things didn't always work as we expected, but I think that we had to be patient, and then the rewards were there. And so I thank you for your attention, and if you have any other questions, I'm happy to answer them. Maybe I should stop.
Thank you very much, Rosa. This was a beautiful journey through the hub neuron discovery.